Hello. Yeah, we are going to be talking about the electron transport chain. After the TCA cycle, we assume that NADH and FADH2 were obtained. So, this is the I assume that this is the mitochondria. Here is the mitochondria matrix, and this is the inner mitochondria matrix. While here is the intermembrane space. So from complex 1 here to complex 4, that's what we call the electron transport chain. While from complex 1 to the ATP synthase is known as the oxidative phosphorylation. While the ATP synthase alone is known as chemiosmosis. So here in complex 1, NADH is what works in complex 1. NADH here gives out its electron to complex 1. It loses electron and proton. So once the electron gets to complex 1 gets supercharged and it now have the ability to pull protons from the mitochondrial matrix into the intermembrane space, forming mass of protons here in the intermembrane space. You can see the proton gradient forming in the intermembrane space. Now that's what works in complex 1. Here in the electrons here in complex 1 also move from complex 1 to the co-Q and stays there. In complex 2, FADH2 is what works in complex 2. It loses its electrons and forms FAD. Once the electron gets to complex 2 cannot get supercharged. So the electron move from complex 2 to the co-Q as well. So the co-Q is a common electron acceptor for both complex 1 and complex 2. Now, the electron that sits in co-Q moves from co-Q to complex 3. Once the electron gets to complex 3 gets supercharged and propels protons from the mitochondrial matrix into the intermembrane space, forming another mass of protons here. Here again, the electron move from complex 3 to cytochrome C or the ubiquinol. Once it gets there, it sits for a while and move from cytochrome C to the complex 4. Complex 4 receiving the electron gets supercharged again, just like what happened in complex 1 and 3. It gets supercharged and move protons again from mitochondrial matrix into the intermembrane space, forming another mass of protons here. So a complex 4 now gives electron to the final electron acceptor in the electron transport chain, which is oxygen. Oxygen receives this electron and then splits into two oxygen ions. On receiving proton, it changes to two water molecules, H2O. So oxygen receiving electron and proton gives two water molecules. Here in the electron transport chain, oxygen is the final electron acceptor. So coming over to the ATP synthase now. ATP synthase is a kind of pump, or you call it the ATP pump. The work is that it propels all these protons in the intermembrane space. It propels them, it brings them back, it draws them back to the mitochondrial matrix. How does it do this? Because technically, molecules always like to move from a region of higher concentration to a region of lower concentration in order to maintain or obtain equilibrium. So ATP synthase here has to drag protons from the intermembrane space down to the mitochondria matrix. Here, that's what brings about ADP here. Now, ADP here is a kind of is a kind of lesser energy source. So this lesser energy source needs to be promoted to a higher energy source, which is the ATP. Now, how does this occur? When the protons move from intermembrane space to the mitochondrial matrix, it moves to the ADP, turning ADP to ATP, which is a higher energy source needed in the body for the various metabolisms going on. This is how the electron transport chain work, or the oxidative phosphorylation work. It continues in order to produce what? Massive amounts of ATP for for metabolic processes. Here, it doesn't just occur. There are certain drugs or molecules that inhibit all these processes. Here in complex 1, we say that the molecule that inhibits complex 1 is what we call the rotenone. The rotenone inhibits complex 1, and then here in complex 3, the antimycin inhibits complex 3. And in complex 4, we have the cyanide. The cyanide inhibits complex 4. And coming over to the ATP synthase now is the oligomycin. The oligomycin inhibits ATP synthase. Why there are kind of molecules again that propels the movement of this proton down to the mitochondrial matrix. They are known as uncoupling agents. Uncoupling agents like the 2,4-DMP helps to pull down all these protons from the intermembrane space back to the mitochondrial matrix. Okay, finally, I have to say the goal of electron transport chain is to couple electron stored in the electron acceptors to a proton gradient that drives ATP synthase. Thanks.